Okay, Chapter 5, Section 3. Protein interactions modulated by chemical energy. Actin, myosin, and molecular motors. Now we need to keep in mind that the cells of your muscles have a cytoskeletal component composed of myosin and actin that allows for the foreshortening of the cell. You get enough of those cells foreshortening, you get a muscle fiber foreshortening. Get enough muscle fibers foreshortening, you get a muscle contraction. And what we're going to be looking at here is discussing how the myosin and actin interact via motor proteins to pull and thus shorten the cell. You look at myosin. Myosin is composed of 2 heavy and 4 light chains of proteins. They're fibrous proteins. Fibrous proteins that the majority of it you see down here are alpha helices, right twin twist. You take those 2 alpha helices and you wrap them around each other in a left-hand twist. Remember, the counter twisting gives a more stable structure. Actin is composed of, G subunits of actin, G-actin, that are just polymerized, and then they are wrapped around each other in a right-hand twist to form the long polymer known as F-actin, filamentous. G-actin's the subunit. String enough of them together, you get F-actin, the filament. You take enough of those myosin and you don't want them individually, because individually they're floppy, so you aggregate them together. You bind them together to form a thicker and thicker filament. And those myosin heads, the heavy subunits, are pointed out, not in towards the center of that aggregate. They're pointing out. Gives areas for proteins to interact with other proteins. The thin filament, so you have a thick filaments of myosin, thin filaments of F-actin. Each of the monomers can bind and interact with ATP, and that's very important here. F-actin, each of those you see there, those long strands, interacts with troponin and tropomyosin. Okay, tropomyosin, troponin. The F-actin you see there also plays very well with, actually hydrolyzes ATP, cuts off that third phosphate for energy. Okay, all right, so what's happening here? Myosin, actin. They are laid parallel to each other. The interaction between the two, as you see here, is via the myosin head. Okay, because remember, the myosin aggregates and those heads are pointed out in all directions. Well, you're going to have myosin and actin stacking around each other, not only in one dimension, but in all dimensions, wrapping around each other within the cell. The direct interaction through the head. What I keep saying is a muscle cell is actually known as myofibrils. Okay? The myofibrils, which are surrounded by what is called the sarcoplasmic reticulum. That's an extracellular version of ER, where calcium is stored. You see that the myofibrils are broken down into subunits known as sarcomeres. Okay, the sarcomeres, cells, long strand of cells, myofibril, get the myofibrils wrapping around each other. You start to get muscle fibers, get enough muscle fibers together. You get bundles of muscle fibers give you a muscle. The sarcomere can span multiple cells, okay? So we're not thinking cell to cell. We're thinking contractile. Contractile to contractile to contractile region. It's all going to be based on those I bands. These I bands are protein structures that the actin filaments are going to be attached to. I band, actin filament coming off of it. I band, actin filament coming off of it. Actin filaments coming to the center. And what crosses between those actin filaments, there's the myosin. Myosin heads interacting with the actin filaments. Okay? Well, those I bands are also have areas known as Z discs. Okay? I'm not going to get into all of the different, because honestly I don't care. Okay? I bands is the region. Z disc is the actual protein. There you go. What happens, as you see at the bottom here, is those myosin heads interacting with the actin. The myosin heads do this. And as they're doing this, they're pulling the actin closer and closer together. The I bands, with the Z disc in the center of them, come closer together. Foreshortened. Remember I said the cell foreshortens, blah, blah, blah. Okay? Sarcomere foreshortens. Get enough of the sarcomeres foreshortening, myofibril shortens, and so on. What's happening here is that for those myosin heads to do this, you have to have ATP hydrolysis. ATP, remember, that's our source of chemical energy for pretty much all metabolic functions. The hydrolysis of ATP, cutting off that third phosphate, brings in negative 7 kilocalories per mole of energy. Foreshortens. Now, this is controlled by different proteins being present. Our friends tropomyosin blocks the myosin interaction. Tropomyosin does not allow the myosin to interact, so the myosin, even if it was to go like this, it's not moving at all, not going to move the actin because there's no interaction. The troponin, in the presence of calcium, changes shape, interferes with tropomyosin. So now, if tropomyosin is interfered with, the myosin head can interact, ATP gets hydrolyzed, goes like this, actin gets pulled toward the center, sarcomere gets... Now, where's that calcium coming from? Well, it turns out that calcium is being released by the sarcoplasmic reticulum. Signal from the brain travels down the spine, jumps central to peripheral, peripheral down to the neuromuscular juncture of whatever muscle you want to contract. That signal jumps from the nerve to the muscle over that synapse, but instead of causing contraction, no. What it does is it tells the sarcoplasmic reticulum to release all of its calcium. Calcium binds to troponin, causes troponin to now be activated, interferes with tropomyosin. Tropomyosin can no longer stop the myosin heads from interacting with actin. ATP gets hydrolyzed. Boom. Actin gets pulled. Is that completely everything going on? No. That is a very remedial, elementary discussion of what's going on, and honestly, that's the level I want you to know it at.